Thank you for the introduction. Can I have my slides, please? It's a pleasure for me to present at this conference on the topic of precision nutrition based on metabolic phenotype for the prevention of metabolic disease. When we look at the prevalence of overweight, obesity, and related diseases like type 2 diabetes and cardiovascular diseases, we see in this slide the prevalence of overweight, which is in most countries of Europe and worldwide 50% or higher. And the prevalence is still increasing, which of course has an impact also on the related diseases. So we really have to go towards more effective prevention strategies. And we know that when we look at lifestyle intervention, it improves postprandial glucose response, it reduces diabetes risk, and may improve our metabolic health. And that's what you see depicted in this slide. Data from the European Diabetes Intervention Prevention Study, where a lifestyle intervention according to general guidelines, diet and physical activity, was given to individuals in the pre-diabetic state. And what you see here in this slide is the cumulative survival free of type 2 diabetes. And when you look at the slide, you see that in the intervention group, this is much higher over the years as compared to the control group. So the cumulative incidence of diabetes and metabolic syndrome is reduced by 57% by lifestyle intervention. When we look a little bit further at those data, we see, however, that 30% of the individuals do not adhere or respond to the intervention, and there's a large variability in response. So the question is, can we optimize the effect of health or lifestyle intervention on health by, for instance, adjusting the intervention to personal characteristics? And then we come in the field of precision nutrition, and here you see a definition: an approach developing comprehensive and dynamic nutritional recommendations based on individual variables, including genetics, microbiome, metabolic profile, health status, physical activity, dietary pattern, food environment, as well as socio-economic and psychological characteristics. So it's increasingly clear that one size does not fit all, but really to personalize advices, we have to include many data related to nutrigenomics, for instance, our gut microbial composition, a profile of metabolites in blood or at tissue level, but also our lifestyle factors, our level of physical activity, our food behavior, and our dietary habits. And that this personalization may be feasible has been shown in several studies. And here you see a study of Savi and co-workers, which is an Israeli group. And what did they do? They determined in 800 people personal features like microbiome, blood tests, questionnaires, anthropometric, food diary. They took all those data together, developed an algorithm, and they tested that algorithm in daily life to predict glycemic responses, and really that appeared to be working, that may be successful. And they were able to successfully lower post-meal blood glucose response and its long-term metabolic consequences possibly. And also another study looking at the postprandial responses to foods is the PREDICT study, which is a well-known study. And what did they do? They determined triglyceride responses and glucose responses after a mixed meal, and they investigated to what extent they could predict those responses. And indeed, I won't go into detail, but we see, for instance, serum lipid markers, microbiome as important determinants of the triglyceride response, and also meal composition, meal context, microbiome, and genetic as determinants of the glucose response. So it's clear that the machine learning model predicted both triglyceride and glycemic responses to food intake. And I show you just these data because those derived data may be informative for developing personalized diet strategies. And another approach, which we also follow within our research group and laboratory, is to base precision nutrition on metabolic phenotypes, metabotypes. And that's what is depicted in this slide. On the left-hand side, you see population-based nutrition. And when you divide that population in subgroups based on metabolic phenotyping, you see here different subgroups on the right-hand side. And for each subgroup, you may define an optimal diet, which may improve health even more than a standard diet. And we focus very much on parameters of glucose and insulin metabolism because it has been previously shown that they may predict the response to dietary intervention. And of course, the degree of insulin resistance, which is a reduced action of insulin within the body, is very important in determining the response to food. Now, we know that insulin resistance may develop in several tissues within the body simultaneously. For instance, you see here adipose tissue, liver, and muscle. But there also are subgroups of individuals which may develop, for instance, more pronounced liver insulin resistance and remain relatively insulin sensitive at level of muscle, or a group that may develop more pronounced muscle insulin resistance. And it has been shown that those phenotypes of more pronounced liver or more pronounced muscle insulin resistance may be associated with a different cardiometabolic risk profile. And you can imagine that also those different phenotypes require organ-specific interventions. Two questions can be asked when you focus on those phenotypes, and I present you this just as an important example. When you look at, for instance, those phenotypes of more pronounced liver and muscle insulin resistance, that's around 1/3 of the population with overweight. And the first question you have to ask, are these phenotypes really distinct? And then when that is the case, is the second question, do those phenotypes respond differentially to diet? And those things we investigated in our research. At first, we would like to see whether those phenotypes are really distinct. And we did, to investigate this, we determined the plasma metabolome, lipidome, and adipose tissue transcriptome profile in overweight individuals with insulin resistance in either skeletal muscle or liver. And I'll show you some data on that. But at first, how do you define tissue-specific insulin resistance? Is it hard to measure that? We did that based on an oral glucose tolerance test, where individuals receive 75 grams of glucose, and after that we follow the glucose and insulin response. So a 7 points OGTT. The blood is collected at 7 time points. And from that curve of glucose and insulin, we can then determine the hepatic insulin resistant index, and that is the area under the curve of the first part of glucose times insulin. But we can also determine muscle insulin sensitivity, which is derived from the last part of the curve. It's the glucose concentrations from peak to nadir divided by the mean insulin concentration. And by having that, we can divide and classify our population in several groups. Individuals with more pronounced liver insulin resistance, that are individuals in the higher tertile of Heery. Individuals with more muscle insulin resistance, that are individuals in the lower tertile of MESI, a combined group, and a group with no insulin resistance. And just to show you some results, what did we do in a large cohort of 500 individuals? We took a biopsy of abdominal subcutaneous adipose tissue, and we can determine in that biopsy all genes by means of gene sequencing, also expression of all genes. And here you see results for liver insulin resistant and muscle insulin resistant, both versus no insulin resistance. And we did a kind of pathway analysis. So what pathways are enriched in either phenotype? And what we did see is that liver insulin resistance was associated with extracellular matrix remodeling pathways, whereas muscle insulin resistance was associated with an enhancement of the abundance of inflammatory pathways. And we validated the latter finding more into detail, we also found in circulating plasma that the inflammatory pathways or the inflammatory score was elevated. And that may be one of the mechanisms why, for instance, adipose tissue inflammation is linked to skeletal muscle insulin resistance. But just in summary of our findings, as indicated, the muscle insulin resistance was associated with an increased expression of genes related to inflammation, in particular related to chemokines and complement system, and an increased level of plasma low-grade inflammation, whereas the liver insulin resistance was associated with an enhanced expression of genes related to extracellular matrix remodeling. And I'm not going to discuss any mechanisms with you. It's of course just to show that those phenotypes are really distinct. And beside the adipose tissue transcriptome profile, we found a very distinct profile of metabolites and lipids in blood in those phenotypes. So those phenotypes are distinct. Then the next question is, do those phenotypes really respond differentially to diets? And that's what we investigated in the person study, which is a large precision study, precision nutrition study, and a kind of proof of the concept to test whether different insulin resistance phenotypes show a differential response to diets. What did we do? We screened a large group of individuals, around 900, did an oral glucose tolerance test and divided them in muscle insulin resistance and liver insulin resistance. Both groups received two diets, either a diet high in monounsaturated fatty acids or a diet in low fat, a diet low in fat, high in protein, and high in dietary fiber. And so both groups followed both diets, and for comparison, we divided those groups in the pheno-diet group A group, where we see the muscle insulin resistant group on the high MUFA diet and the liver insulin resistant group on the low fat, high protein, high fiber diet. And pheno-diet group B, where we see the liver insulin resistant group on the high MUFA diet and the muscle insulin resistant group on the low fat, high protein, high fiber diet. So just the other way around. This was a large study performed at Maastricht University and Wageningen University within the context of a large public-private partnership within TIFFN, the Top Institute Food and Nutrition. And what we did, it was a 12-week intervention, an isocaloric intervention, and before and after we did a very detailed phenotyping in all individuals. And that's what you see here. For instance, we determined body composition and ectopic fat, that is fat that accumulates in other tissues than adipose tissue. We determined cardiometabolic risk profile. We did challenge tests with an oral glucose load or a high fat load. We took questionnaires, and we did deeper phenotyping related to biopsies and clamp methodologies. Over the period of the 1 week, we did continuous glucose monitoring to determine glucose profile. We mapped physical activity and, of course, the dietary food intake. And here you see the two diets we used, as indicated, the high MUFA diet, which is high in monounsaturated fatty acids, 20 energy percentage, and the low-fat, high-protein, high-fiber diet, which is lower in fat content, higher in protein content, and that was half animal-based and half plant-based protein, and relatively high in dietary fiber, so more than 4 grams per megajoule per day. So I'll show you some of the results of this study. And again, here you see the Meer and Lear group and how it was divided in the phenotype diet A and B group. First, some results of the disposition index. And what is the disposition index? That's a composite marker of insulin secretion adjusted for insulin resistance. And here you see the line before and after intervention of the pheno-diet group B, and here you see the pheno-diet group A. And there were no differences or only a slight tendency between groups with respect to the disposition index. But when we look further at insulin sensitivity or insulin resistance, there were pronounced differences between groups. In general, the phenotype diet group B did better than the phenotype diet group A. And just to refer back to that, the phenotype diet group B was the muscle insulin resistant group on the low fat, high fiber, high protein diet, and the liver insulin resistant group on the high MUFA diet. And we did see that the HOMA-IR, which is a surrogate marker of insulin resistance, decreased more in phenotype diet group B as compared to phenotype group A. The same picture we see for the Matsuda index, and Matsuda index is a measure of whole body insulin sensitivity based on the oral glucose tolerance test. We see a more pronounced increase in the phenotype group B as compared to A. And again, here you see the combination, the MER group on the low-fat, high-protein, high-fiber diet, and the LER group on the high-MUFA diet represents the phenotype diet group B. The same we see for MISI, and MISI stands for the Muscle Insulin Sensitivity Index, as indicated before. It increased much more during intervention in the B group as compared to the A group. And when we subsequently look at post-hoc testing, where we divide it into MER and LER and the two different diets, you clearly see the HOMA decreases more in the MER group on the low-fat, high-protein, high-fiber diet, and in the LER group on the high-MUFA diet. The same picture we see for Matsuda index, a more pronounced increase in the MER group on the low-fat, high-protein diet and the LER group on the high-MUFA diet. And the same picture arises for the muscle insulin sensitivity. So in general, the phenotype diet group B appears to do much better with respect to parameters of insulin sensitivity, and in particular peripheral insulin sensitivity. And the increase is 20%. So that's really a clinically relevant increase when individuals receive the diet according to phenotype diet group B. Then we come to results of serum triglycerides, and we saw actually a same picture with respect to that, a higher decrease in phenotype diet group B as compared to A. And when we look at the post-hoc testing, it seems to be merely determined by a higher decrease in triglycerides in the MER group on the low-fat, high-protein, high-fiber diets. When we look at the inflammatory marker CRP, again, we see a more pronounced decrease in the phenotype diet group B as compared to A. And again, you see a more pronounced decrease in inflammatory marker CRP in the MER group on the low-fat, high-protein, high-fiber diet and the LER group on the high-MUFA diet. We also looked in detail at body composition, body fat, visceral adipose tissue, and ectopic fat, and both groups or phenotype diet group improved with respect to that, but we did not see any differences between groups in body fat percentage, visceral adipose tissue mass, and liver fat percentage. And hepatic insulin resistance, we did not see any differences between groups, and that refers again back that we saw an improvement in insulin sensitivity, but that was merely an improvement in peripheral insulin sensitivity and not at level of the liver. So in conclusion from that study, modulation of dietary macronutrient composition according to insulin-resistant phenotypes results in pronounced clinically relevant further improvements in cardiometabolic risk markers, and that was independent of changes in body weight. And this is one of the first double-blind randomized trials to provide indication that precision nutrition for metabolic subgroups may be superior to a diet based on general guidelines, healthy nutrition for improving cardiometabolic health. So both diets were healthy and they were within the boundaries of guideline healthy nutrition, and both diets results in an improvement in metabolic health. But when we adjusted or modulated the diet according to phenotype, the improvement in metabolic health was much more pronounced, around 20% more pronounced. And of course, these findings are pronounced, and they may provide additional information for future strategies in the field of precision nutrition. But it's also important that we understand why people respond differentially to diet. And of course, we did a very detailed phenotyping also in the person study, and we're digging currently also in the underlying mechanisms. And what could be an important factor playing a role when you divide individuals in subgroups, you have their metabolic phenotype, but also the microbes in our guts may play an important role in the interaction with diet, and may, for instance, be an important determinant of the intervention outcome. So our idea was there may be an interaction between the microbial phenotype and metabolic phenotype, which defines whether people respond or non-respond to certain diets. And as indicated, there are indications in literature indeed that the baseline microbiota composition, so the baseline composition of the gut microbes within our guts, of our gut microbes, may be an important determinant of intervention outcome. And here you see some studies related to that. The first study is a study where a fecal transplant took place. The feces of lean donor individuals were transplanted to individuals with the metabolic syndrome, and a pronounced transient increase in insulin sensitivity was found in that study, but it appeared to be driven very much by baseline microbial composition. So that may be very important in the extent to which individuals improve in insulin sensitivity. And also another study, and this is with prebiotic supplementation, the idea was to study effects on gastrointestinal transit and metabolic health. And overall, at group level, we did not see that much effects. But when we relate the overall microbial composition to the study outcome with respect to, for instance, gastrointestinal transit, the microbiota composition appeared to be an important determinant of outcome. And of course, there are more examples in literature that microbial composition may be important in the diet response. And for that reason, of course, we looked also in our phenotypes in the person study in Meer and Lear, whether there could be differences in microbial composition. And what you do see here is the Bray-Curtis index, which is an index of beta diversity. And the most important thing, and that I would like to emphasize in this slide, when you look at the Meer group in purple or blue and the Lear group in orange, you see a clear clustering of phenotypes. So the microbial composition was different in liver and muscle insulin resistance. And when we looked at measures which also reflect the function of the microbiota, like short-chain fatty acids, you see here the concentration of short-chain fatty acids, acetate, propionate, and butyrate in feces. They appear to be higher in the liver insulin resistant group as compared to the muscle insulin resistant group. Also, when we looked at, for instance, plasma GLP-1, and plasma GLP-1 is importantly driven also by microbial composition and the production of short-chain fatty acids. And when we studied that after a high-fat mixed meal, the level was much higher in liver insulin resistant as compared to muscle insulin resistance. And when we look more specific at different genera or taxa, you see that depicted here, a higher abundance in the liver insulin resistant group of taxa involved in short-chain fatty acid production, whereas that was not the case in the muscle insulin resistant group. So in general, the Lear group, the liver insulin resistant group, appears to be associated with a gut microbial profile related to short-chain fatty acid production, and also higher levels of short-chain fatty acid production in feces, higher GLP-1 responses, so may represent a more healthy microbial phenotype. And of course, we're currently determining to what extent that also determines the dietary intervention outcome. We have the idea that it may be the case because we previously showed that in particular the production of short-chain fatty acids by gut microbiota may impact metabolism to a large extent. For instance, we administered through cannulas clipped to the gut wall by endoscopic techniques. We administered short-chain fatty acids either at the more proximal colon, and we did not see any metabolic effects. But when we applied those short-chain fatty acids or administered those short-chain fatty acids at the distal colonic site, we did see very pronounced metabolic effects related to fat oxidation, related to a reduced inflammatory profile and a reduced lipolysis. So those effects appear to be beneficial, in particular when they translate into the long term. And also in those conditions of distal colonic short-chain fatty acid administration, there was an increased concentration of circulating short-chain fatty acids, which may be an important driver of the effect. So, and that was what we specifically also investigated in this study. We looked at dietary fiber combinations which increased the distal colonic fermentation and short-chain fatty acid production. And we first screened a lot of fibers in the in vitro model of the colon, the TIM2 model, and we selected fibers and subsequently administered them acutely in vivo to humans. And we administered them from one day, and the next morning, we studied them and performed a meal breakfast test. And what we did see when we administered, for instance, inulin resistant starch, which was very effective in increasing distal colonic short-chain fatty acid production, we saw an in vitro increase of short-chain fatty acids. In vivo, we saw an increase in hydrogen breath, so indicating increased fermentation. That's just a crude marker. But we saw an increased plasma butyrate, these short-chain fatty acids, an increased energy expenditure, an increased insulin sensitivity in the postprandial state, an increased postprandial PYY concentration, and an increased fasting butyrate in plasma. Whereas when we looked at individuals with overweight and pre-diabetes, actually we did not see any effects, no increase in short-chain fatty acids, but also no increase in metabolic health effects. So, and what is the case that the effects were fiber-specific. We only saw that, for instance, with inulin-resistant starch, whereas there were no effects with other fibers like beta-glucan and resistant starch. So we have the idea that the balance between carbohydrate fermentation and protein fermentation and the related products you see indicated here may be very important for metabolic health. With carbohydrate fermentation and the related short-chain fatty acids having in general a more positive effect on tissue level, on beta cell function, muscle metabolism, the storage capacity of adipose tissue, and inflammatory profile, whereas in general protein fermentation may have more adverse effects. So the balance between dietary fibers and also proteins within the diet may be an important determinant of health. The take-home messages. Distinct tissue metabotypes or phenotypes can be identified towards type 2 diabetes. The dietary macronutrient modulation according to these metabotypes substantially further enhance cardiometabolic health, and that's independent of changes in body weight. LIR and MIR and liver and muscle insulin resistance are characterized by a differential microbiota composition, with LIR having more short-chain fatty acid producing taxa and increased fecal short-chain fatty acid concentrations. So variation in microbial composition and functionality, in particular fermentation, may affect tissue dysmetabolism differentially and may thereby mediate the dietary intervention response in tissue metabotypes. So dietary intervention success in tissue metabotypes may relate to the balance between the saccharolytic and proteolytic gut microbial fermentation. And I would like to end my presentation with thanking all the people that have been involved in the research, and also by thanking the funders of this research, in particular the companies related to the TIFFN project, the Person study, FrieslandCampina, Nutricia, and DSM. Thank you very much for your attention.